Hi. We might as well start. I'm Pete Schultz. I'm CEO of Scripps, and it's my pleasure to welcome everybody, those here in person and those on Zoom, to our annual John C. Martin Memorial Lectureship. And I'll just start out by saying a few words about John, who I was lucky enough to know. John really, through his life's work, had a huge impact on really the way we treat viral diseases and really transformed the treatment of those diseases, not only in the U.S., but globally. John was remarkable, and remarkable not just as a scientist, but also as a business person and also as a humanitarian. And normally in the world, you meet a person who did one of those things well, or maybe even two. You're a great business person and humanitarian like a Bill Gates. But rarely, if ever, do you meet somebody who does all three of those things at a remarkable level. And that was really John and John's impact. And because he was one of those rare individuals that did science, business, and global benefit well, I think he's perhaps one of the best role models we can have for young scientists today. He was certainly a role model for me in my career. John's scientific contributions really were pioneering the use of nucleosides and nucleotides as antivirals. That started with the work for the first treatment of CMV with ganciclovir, and then he went on with nucleotides, and they developed tenofovir, and then really the single pill treatment of HIV, which really transformed the treatment of HIV. And those achievements led to many other antivirals and impact on the antiviral, on the fight against viral disease. John also, as a businessman, didn't found Gilead, but transformed Gilead into probably the world's leading antiviral pharmaceutical company, and one of the few that grew from inception to being one of the largest in the U.S. And I actually looked up, and over John's, I think, 20-year tenure as CEO of Gilead, its market cap increased 140 times. So if you were lucky enough to be there early on, you would understand the significance of that. As a humanitarian, John really was one of the first people to lead the effort to make low-cost medicines available to low- and middle-income countries. And through those efforts, John and his remarkable group of colleagues at Gilead really saved millions and millions of lives and continue to save millions of lives to this date. And we were lucky enough to have John on the board of Scripps. He was a real friend of the Institute and was on the board until his untimely passing. He was terrific because he was one of the few people on a board that could actually bridge basic research and great basic research with translational research and with, business savvy to understand how best to move the Institute forward with resources it needed. And John was always there with really terrific insights and guidance in a really terrific way from a CEO perspective. He never pounded the table. It was like, Pete, maybe you should think about this. So it was really a great experience to work with John to build Scripps. And I can't count the times that people sit around a table and say, I wonder what John would have thought about that, or it's too bad John's not here today to help us out with that. So a huge impact on the Institute and the vision for Scripps. That legacy is continued today with Lily, his partner, who serves on the Scripps board, and we again appreciate her guidance and insights greatly. With their collective philanthropy in funding graduate fellowships. John was always a big fan of graduate students. Chairs for faculty, and also some of our efforts in developing drugs for unmet needs in the developing world. And also, we benefit from our relationship with John because of the involvement of some of his ex-Gilead colleagues and Scripps. In particular, Bill Lee and Tyann and Norbert serve on our scientific advisory board and give wonderful advice and generously of their time to help us move programs forward. So not only was there John's impact on the world of science and on global health, but also on our own institution, which continues today. So it's really terrific to celebrate his life and his accomplishments. We do that. John had really two unique aspects to his scientific life, and that was his interest and enthusiasm for basic research. And so we have a speaker today who reflects that, Stuart Schreiber. Also, his interest in translational research, and likewise, we have Nick Leeper here today to talk about translation. And then kind of the impact on science on human health and around the globe. And we have Greg Alton here, I think, who's going to finish up with remarks that underscore John's contributions there. So again, it's a pleasure to have you here and to have this lectureship to honor John's accomplishments and memory. And I think with that, I'll ask Lily to introduce Nick. Lily? Yeah, I didn't know it either. Do you want to? That would be great, Lily. Thank you. That's impromptu. Very good. And then I'll have Ben follow with some impromptu remarks about Stuart.
For the way I'm dressed so casually, and I have no recollection of where Nick went to school or got his training. But at any rate, you can look all those things up online. I'll just say a few quick words about how after John's left Gilead, he started happily and excitedly his new second life. And Nick, you were one of his newest BFF. So, Nick, your research, I'm sure you're gonna go over this, started with anti-CD47, and then to skip all over that, John, you approached John and you met him when you formed your company called 48, and now it's called Bitterroot. And Nick is gonna tell you all about the fantastic translational research that's gonna save a lot of people's life. And I can just say that because of your fantastic research accomplishment, he was so interested and really, really enjoyed working with you, and the friendship that comes based on the mutual enthusiasm about science, medicine, and saving people's lives is what bonded you together, although the time was very short. So thank you for joining us, and I hope that these are the opportunities where friends from different corners should come together and make new things happen. And that's what John would do, like bring people together so that because of your mutual interests, you make more things happen and we make new memories to bring well-being to people who need help. Thank you.
Well, Lily, thank you so much. If that was impromptu, that was really remarkable. And thank you to Dr. Schultz, and thank you for having me at this incredible lectureship series. As mentioned, my name is Nick Leeper. I'm a professor of medicine and surgery at Stanford University. And I'm really excited to share a real translational story today about our work. And it's really particularly touching to me, at least, because so much of this was done with John. So I hope you don't mind if I pepper in some stories as we go. Just by way of disclosure, I'm the co-founder of 2 companies, and I will be discussing some of the work that was done through these organizations in today's lecture. But the most important disclosure is, this is not working. Don't know if I can get some help advancing the slides. Okay. This is the most important disclosure. I like to start recognizing the team that did the work, and today it's really extra important and special to me because 2 of my former trainees who are now on faculty at San Diego, including Elsie Ross, surgeon scientist, and Tom Alsay, they're in the crowd, are here. So I just want to say thank you up front for all the work that you've done to make this story possible. But for me, this is really special because John was a giant, as we heard, and an incredible mentor to me. I learned so much about him, and even though I've been at Stanford for more than 20 years now, I trained at UCSF, and I remember the days very clearly when HIV and hepatitis C, and these were death sentences, and through so much the pioneering work and the vision that John brought to this, these are manageable, if not curable, conditions in several regards. And so, I hope that you'll, sense my emotion about how fortunate I feel to have gotten to work with and learn from John. And I'm just so happy that his memory and legacy lives on in this venue. But a really special thank you to Lily because of her continuing friendship since John's passing. And so, Lily, thank you for inviting me. This really means a great deal to me. So the story I'd like to tell today is one about the type of problem that I think John enjoyed trying to conquer. And in this case, this is our work trying to address cardiovascular disease, which, of course, is the world's leading killer. Our lab is focused on atherosclerosis, which is the process underlying heart attack and stroke. It's important to remember that if you add up all types of cancers, HIV, AIDS, accidents, all that combined claims fewer lives than this phenomenon. And so this is what I set out to work on when I began my career. And when I was a trainee and a postdoc at Stanford, my mentors, really challenged me to think about the current paradigm and what we might be able to do differently. And they pointed out to me that if you think about how we treat heart disease in today's modern era, we've gotten very good at addressing risk factors for disease like blood pressure, smoking, and cholesterol. A lot of innovation around antithrombotics to prevent clots from forming on top of the plaque and helping people with heart attack and stroke. But at the end of the day, most cardiologists are plumbers, and we have a lot of fun in restoring the mechanical problems and improving blood flow by putting in stents or doing bypasses. But one could argue that we do very little to address the pathobiology in the lesion itself. And this seems like a real missed opportunity for us, especially given that this still is the leading killer. you could argue that it doesn't matter because cardiology as a discipline has had decade after decade of steady improvement. And you can read articles like this where they'll point to several of these breakthrough discoveries and say, well, we're on this downward trend, and so what are you really worried about? But you could also take a different perspective and argue that most of this improvement is really due to just two phenomenon, including the change in smoking behavior in the Western world and the advent of statins and lipid lowering. And it's fine to put other things on this map, but I would argue that we may be getting to the point of diminishing returns, and that as lipids are driven lower and as smoking becomes less and less common, we are going to plateau on this curve. And indeed, we have started to see this. There's actually been an uptick in cardiovascular disease, due in part to the obesity epidemic and other metabolic disorders, but also due to diastolic heart failure and people surviving their myocardial infarction only to go on to develop ischemic cardiomyopathy and other forms of heart failure, which are now the leading discharge diagnoses in the United States. So we've seen this uptick, and I, for one, am very interested to see what the GLP agonists do to this curve. But at least with today's state, this is still, as I mentioned, the most pressing public health issue on a per capita basis. And this is where I have been disappointed with many of the large pharmaceutical industry efforts out there, because they, in my estimation, have continued to focus on the low-hanging fruit and to drive the established risk factors lower and lower. And there's no problem trying to get your LDL cholesterol down lower, and we've seen tremendous benefits with things like PCSK9 inhibitors and other lipid-lowering therapies. But you can also, take away the zoomed-in plots and focus really on the absolute risk reduction and see that, these landmark trials that get published in the New England Journal are really showing fairly modest improvements on a number needed to treat basis. And I think this is why you can see all of what we call the residual risk, where even in these trials, which are all wildly successful by modern standards, with nice relative risk reduction, I'm very focused on the pink and the unmet need that we currently are not addressing by lowering blood pressure, cholesterol, and focusing on diabetes. So this begs the question of what else is out there, and I think many in this room will probably be well aware of, in the cardiology field, what we talk about often is the inflammatory hypothesis of atherosclerosis. And really beautiful work by Paul Ridker and Peter Libby and others has set up this kind of idea that there's two key pillars, and there's the cholesterol risk and the inflammatory risk, and these need to be addressed in parallel. And there is actually very beautiful observational data where we can see that lowering your LDL cholesterol is of course important, but if your inflammatory risk doesn't come down, which can be estimated based on things like high sensitivity C-reactive protein, even if your LDL level has fallen but your CRP stays high, your risk doesn't come down. So we have to find ways to not only lower cholesterol, but also get at this inflammation that we think is driving so much of disease. Now, a lot of this is, observational and theoretical, and so people in my field are really thrilled when this landmark paper was published almost a decade ago now. This is the CANTOS trial, where they were able to inhibit an inflammatory cytokine, in this case interleukin-1 beta, and they showed that this blocking antibody would really beautifully suppress inflammation, at least as measured by C-reactive protein. And what I loved about this was that they proved beyond a shadow of a doubt that this was happening independent of any other risk pathway. There was no effect on cholesterol or blood pressure, et cetera. And they proved the hypothesis to be correct. They reduced myocardial infarction. They improved long-term cardiovascular outcomes. But the reason that the clinicians in this room aren't prescribing this every day for heart disease is because this had an Achilles heel. And we know that this was kind of a blunt tool, and in suppressing the inflammation, you were also causing immunosuppression, and there was a significant increase in sepsis and infections, and also death to fatal infection. And so it proved the hypothesis, but it also pointed out the limitations of our current approach. We haven't given up on inflammation, and there are a number of very elegant and genetically informed trials underway. I would say that, and in fact, several of them will report out this year, there is a lot of hope that going higher or lower in the inflammasome pathway may provide that benefit that we desire without the side effects. We will soon know if inhibiting the inflammasome or IL-6 will provide that protection. But I, for one, won't be all that surprised if we continue to see some infectious risk with these very ancient and conserved pathways that obviously were evolved to help us fight off infection. So this is kind of depressing, and the question is, okay, well, what else can we do? And again, my mentors taught me early on, and I know Tom and Elsie have seen me talk about Churchill quite a lot in my talks. How can people do everything wrong and somehow escape their fate, as it were? If you think about Churchill, born in the 1800s, overweight, smoked, drank, one of the highest stress jobs you can imagine, he didn't have his first stroke until he was in his mid-90s, for example. And the converse is a person like Jim Fixx, who is credited as kind of being the father of the fitness revolution in the United States, wrote this New York Times bestseller. It's a picture of his leg on the front of the book to show you just what a specimen he was. He'd go on talk shows talking about, if you can exercise, you can live forever. And, as the story went, he was found dead on the side of the road in his 50s while out running, and was found to have severe 3-vessel coronary disease. So what is it about these people that are kind of tricking their risk profile and then leading to the clinical phenotypes that we wouldn't have otherwise anticipated? And so this is why I just have had so much fun in my academic career, because I happened to come through my training right at the era of genomic medicine and the ability to move from hypothesis-driven approaches to unbiased and hypothesis-free science, particularly with the cost of sequencing coming down, and now the ability to scan the full genome and be surprised and not go in thinking that my gene or my pathway is the most important, but let's see what biology will tell us. And so I worked with some fantastic geneticists, including the late Jeanette Erdman here. This is one of her review articles from, again, almost a decade ago now. And at that time, there were 163 loci that were kind of irrefutably associated with risk of coronary disease. And I really like this because you can rediscover things that you expect, like the lipid metabolism and blood pressure on the top, but look at how many other things we were able to discover that you might not have anticipated if you'd gone in reading textbooks from when I went to medical school. We had no idea about the role of cancer genes in heart disease. We kind of thought that neovascularization was important, but not really a key driver. And then I love the bucket at the bottom, all these unknown genes, which means that I'm going to have job security because we have a lot of work left to do to figure out how these things are causing disease and then how to target them. And so, my personal journey and the story that I'll tell you today began, again, just right when my clinical training was ending and I was coming in the lab. There were these beautiful genome-wide association studies that were published almost simultaneously by these 3 groups, and they all came to the same conclusion. And it was just a remarkable kind of renaissance for us because we now had a smoking gun that the field could go after. Now, for those who haven't seen a GWAS result, this is what's called a Manhattan plot. Each chromosome is laid out on the x-axis, and then what you'd like to see is when a genetic variant is more commonly found in people with disease compared to without. The stronger the association, the taller the skyscraper. And the punchline for these papers was that each of them came to the same conclusion that the most important heritable locus for coronary disease and myocardial infarction is found at chromosome 9p21. And you just love seeing, a finding like this. Now the p-value is 10 to the minus 200th or something. And when I read these papers, I was floored. First, because these are not rare mutations. These alleles have a very high allele frequency. It's been estimated that about 20% of the population carries 2 copies of this, so 1 in 5 people in this room has this. This is what they used to test if you had a 23andMe test, for example. And then the other thing that really jumped out was that this locus was 100% independent of everything we knew about. If you were born with these risk alleles, your risk went up whether or not you were a smoker, whether or not you exercised, whether or not you had diabetes, whether or not you had high blood pressure. And so, I said, gosh, if we start to work on this and we can figure out how this is happening, this will be orthogonal. This will not be yet another blood pressure pill or cholesterol pill. So my mentor had me make knockout mice for all of the genes at this locus. This actually resides in a gene desert, and believe it or not, to this day, we're not entirely sure which gene causes risk at 9p21. I'll spare you some of the details, but we had reasons to believe that actually a cancer gene called CDKN2B, which regulates the cell cycle, was likely to be the causal factor. And when we unblinded the first experiment, we saw, as predicted by the genetics, that yes, these mice had larger plaques. And for those who aren't familiar, this is oil red O stain. It stains lipid red, basically. On the top is the aortic sinus near the aortic valve, and then those are the aortae pinned out. And so this is the other thing that's fun about genetics, is that you usually know the answer before you do the experiment, so less frustration for the postdocs in the lab. But we saw the results, and we thought this was great. But what really jumped out to us was not that the plaques were just larger, but it was the phenotypes of the plaque. And by that, I mean, here is the control group, and this is what you normally will see in the plaque, where you have a lot of collagen and a lot of cellularity, and there's a fibrous cap that we think prevents the plaques from eroding and rupturing. Whereas the knockout mice had these plaques that were almost completely replaced with all of that open area in the middle, and that's called the necrotic core. And this is basically the graveyard of the plaque, where all the apoptotic and inflamed and necrotic tissue will accumulate. And this is what you don't want to see in humans, because this is where all the inflammation comes from. And so we began to wonder if the mechanism by which this was causing heart disease, independently of cholesterol, blood pressure, et cetera, might be an inability to handle all of this waste, and there was a problem taking out the trash. And so we came across this concept of efferocytosis, which is from the Latin meaning to carry the dead to the grave. Now, this is just a 25-cent word for phagocytic removal, typically by macrophages. But as we started to read about this, we came to understand that this is actually a very tightly regulated and finely tuned process. So normally, when a cell starts to commit to programmed cell death and is beginning to die, it will secrete chemoattractants. And these are called find me ligands. And this will recruit the macrophage to the site of injury. This is very rapidly followed by upregulation of what are called eat me ligands. And these are basically cell surface molecules that will interact with receptors on the macrophage. And when this interaction goes well, the corpse is rapidly ingested and degraded. This happens over a matter of seconds to minutes. And then the key thing here is that when this goes well, the macrophage will then secrete a number of anti-inflammatory factors like IL-10 or TGF-beta. And I like to imagine that this is the cell's way of saying, I've got the problem under control, we don't need the cavalry, and the danger signal has been contained so that you don't have ongoing inflammation. The other thing that's really interesting about efferocytosis is that this is highly conserved, and the body is very careful about getting rid of these dying cells and not getting rid of healthy cells. And the problem here is that if a cell is not rapidly removed, the cell membrane will break down, you have secondary necrosis. So this process happens almost instantaneously. This is why the pathologist in the room will remember that it's really quite hard to find evidence of a free apoptotic cell anywhere in the healthy body. If you want to find an example of an apoptotic cell, like these red tunnel-positive cells, you have to look in very high turnover organs like the bone marrow, the thymus, or the tonsil. And occasionally you can find a picture like this where there is an apoptotic cell in red, but you'll notice it's already surrounded by these macrophages in blue. So I like to imagine that you just happen to have gotten lucky. You caught this guy in the act of being eaten and removed, and if this biopsy had been done 5 minutes later, these cells would probably be gone and you wouldn't see any apoptotic tissue. It turns out probably the only exception to this in the healthy body is in the atherosclerotic plaque, which, by the way, we all have been developing since our teenage years. And in the plaque, it's quite easy to find evidence of apoptotic cells kind of floating in the breeze that are not being recognized for removal for reasons that were unclear when this study was performed. Now, they looked at the kinetics of this throughout the body, and they estimated that efferocytosis may be impaired more than 20-fold in the atherosclerotic plaque. But as I mentioned, there were really no clear understanding of why this was not happening well in our blood vessels. And then the final thing about efferocytosis is this point about inflammation resolution. So it's not just about the early inflammatory signal, but now there's a lot of interest in the behavior of the macrophage after the danger signal has been taken care of, and then the switch to resolution, which is very important for tissue repair and regeneration. And so this is all what will happen normally when the process is going well. So I'll spare you this because most of the signaling work is now published, but we went on to show that actually, again, this very interesting cancer gene that is now tightly linked to heart disease regulates a process which will lead to the upregulation of calreticulin. And calreticulin is kind of the granddaddy of all of the eat me molecules. And when that is expressed normally, it will allow these cells to be recognized and removed. But in people who are born with this 9p21 risk allele, we think, and we've shown in humans as well, that they have an impaired upregulation of calreticulin. This renders the cells almost invisible to the macrophage, and then that secondary necrosis will occur when the cell is not removed because the cell membrane has broken down. And it's the release of that necrotic debris which allows the plaque to grow, but perhaps more importantly, to become inflamed. And so this concept has now started to work its way into the textbooks. Often people will talk about it as being the reason that the plaque will physically enlarge over time, but I like to imagine that it's even more important in its role in the inflammation that will destabilize the plaque, ultimately leading to plaque rupture, and then that's what causes the heart attack or the stroke, which is what we really care about. You know, having plaque, that just means you get angina when you're shoveling snow, but plaque rupture is what causes the clinical events. So another fun part about the story and the mentorship that I've been so lucky to have is that the first time my postdoc and I showed the work was at a meeting in Japan. It happened to be near the cherry blossom season in Kyoto. There's this beautiful place called the Philosopher's Trail. And you know, as a junior faculty, I was very proud that we had made this signaling discovery. But the chair of the Nobel Committee said, well, okay, that was fine. Now what are you going to do? And I didn't even think down the road, but he said, now it's time to translate. And so this was one of the first times that I was encouraged to think about how we could reactivate efferocytosis. So my lab spent a lot of time fooling around trying to increase that eat me signal, calreticulin. In retrospect, that probably wasn't very smart because, it's hard to turn genes on in the first place. It's even harder in a cell that's already diseased and inflamed and starting to commit to programmed cell death. And so we eventually came to the concept that perhaps instead of trying to step on the gas, we could instead try to take away the brake. And as I mentioned, the body is very careful about what it does to regulate the removal of unhealthy cells, but sparing healthy tissue. And so almost all of these pathways will have a yin and a yang, they'll have a gas and a brake, and the counterbalance to calreticulin is this very important don't eat me molecule called CD47. So at this point, nobody had looked at these molecules in cardiovascular disease, and we wondered if these molecules are even present in the first place. We started by partnering with the rapid autopsy service at Stanford, and the first coronary artery we got was from a young person who had died in a motor vehicle accident. And what you see is that there's very little expression of these don't eat me molecules in the healthy blood vessel. But in the individual who came in later, who had died of a myocardial infarction, we saw a very intense upregulation, and you can see that that red staining is really co-localizing near that necrotic core where all the inflammation is occurring. We went on to confirm these protein findings at the RNA level, now with that person from the Karolinska Institute who I'd met in Kyoto. He let us look in his biobank, and we found that indeed CD47 was upregulated in both their discovery and validation cohorts. And then over time, we were able to show that this process is gradually turned on as heart disease gets more severe. And it doesn't just correlate with the presence or absence of disease, but also correlates with symptom status. And so patients coming in with a TIA or a stroke will have higher levels than people who have just an asymptomatic plaques. And so this really is starting to not only have these strong links to the development of disease, but also these destabilizing clinical events. And so once again, I won't walk through all of the published signaling studies, but we found at the end of the day that this is yet another link to inflammation, in this case TNF-alpha, which we have shown will drive signaling to express CD47 on these cells. I like to think of this as the cloaking device that prevents these cells from being recognized for removal. And we think this not only allows these cells to accumulate, but then also promote the inflammation. So the next question is if we can target this in vivo for therapeutic purposes. And the other theme that I came to learn early in my junior faculty career was the power of interdisciplinary research. And so it just so happened that around the same time, the oncology doctors were starting to realize that the very same pathway is almost universally turned on by cancer. And this is a mechanism by which tumor cells will escape immune surveillance and avoid removal by the tumoricidal macrophage. They had begun to hypothesize that interfering with this axis might be sufficient to reactivate tumor killing and turn those tumor-associated macrophages into the inflammatory and phagocytic type of cells that are necessary to contain the tumor. And then again, just with serendipity, it turned out that one of the leaders in this field was Irv Weissman, who many of you will know. He's a famous stem cell biologist and oncology researcher at Stanford. And his group had begun to show that whether given alone or in combination with other anti-cancer antibodies, in humanized mouse models, they could reduce or even sometimes cure a wide range of tumors. And so again, I had this idea that I should just try to collaborate with as many people as who were willing to talk to me. And I'd never met Irv, and as I was wont to do in my early days, I wrote a really long email that I think ChatGPT would now make much more succinct. And I wrote this long missive to him asking for a meeting to collaborate, and then I heard nothing. And I thought about giving up, but I waited a couple of weeks, and I wrote him again, and then I heard nothing. But eventually, I did get a response, and I think to this day, although he won't admit it, I think the only reason is because his neighbor, where he goes fishing in Montana, has the same last name as me. I've got no family there. But I happily said, I think I might have a cousin in Montana, Irv. Yeah, so anyway, I got my foot in the door. And this led to a really wonderful collaboration with Irv that translates through the rest of the story. So together with Irv, we set out to see if we could, in fact, bend the curve on atherosclerosis. And we used his tool compound, and we saw that, looking under the EM on mice treated with control antibody, that there were all of these examples of macrophages in the plaque that were just ignoring all of this apoptotic debris. But after giving an anti-CD47 therapy, we saw that we could really rev up the appetite of these cells, and they would take up dozens of targets, and we could confirm this by looking at other forms of apoptotic cell clearance in vivo. When we did this and we revved up the appetite of these cells, we found that the plaques were much smaller. And again, here is this oil red O stain showing how much less plaque is building up near the aortic valve. We saw the same thing throughout the entire body in the aorta. And then again, thinking about translation, we took this further into a plaque rupture model mice don't actually have heart attacks, so you have to find other ways to simulate a plaque rupture. Here we can manipulate the carotid artery, and when you do this, you'll get plaque hemorrhage about 3/4 of the time. That's the red cell stain on the right. That's indicative of the plaque fissuring and likely would be the equivalent to a person having a stroke. And we saw that our antibody could also reduce the plaque hemorrhage, and then lead to stabilization of the cap. So there was more thickening and smooth muscle content. So we think an overall benefit shown there. And so now we came back to our concept that this defect is present. We think it's present in everybody. We think it's especially present in people born with this genetic predisposition for heart disease. But we now had murine data suggesting that it's not a fixed defect and that we could perhaps intervene on this as a way to reactivate the immune system and even shrink established plaque, which is really hard to do in mouse models. And I won't go through these findings either, but we've now branched this out into other areas. It turns out the same phenomenon may have a role in aortic aneurysm disease. And we've shown in multiple mouse models that this could be important for individuals with AAA, for example. Dr. Schultz, we talked at lunch about cardiomyopathy and wound healing. And so it turns out that this isn't just about removal of plaque cells, but also the myocardium that will die after a heart attack. And so multiple groups have shown this can prevent the remodeling and ischemic changes that will happen here, leading to fibrosis. And then work that Tom did when he was in the lab, really, I think, quite elegant work, trying to find Trojan horse nanoparticles where we can deliver high concentrations of pro-ephracitic therapy specifically to the inflamed macrophages as a way to get the benefit where we want it without some of the off-target hematological toxicities that were known to be occurring. And we showed that this would work in mouse models. And then when Tom was in the lab, he worked on these mini pigs. You can see there's nothing mini about them. And in fact, they have these genetic mutations where they will develop atherosclerosis much like a human. And in Tom's work, they showed that this can suppress the inflammation in the arteries of these animals. And so, so that work is ongoing as well. But for today, the story really is about how do we get into humans, and can we make this an actual therapy for people with cardiovascular disease? Now, I mentioned that I was involved in an oncology company, and this group was one of the first to make a humanized anti-CD47 antibody. And this was the first paper published by this group. And this was a non-randomized, non-placebo-controlled study. But the reason that it got into the New England Journal was because these individuals were really kind of at wit's end. They had recurrent non-Hodgkin's lymphoma. They had failed multiple lines of therapy, kind of really out of options, if you will. And at least in this phase 1 study, there were these remarkable changes on PET-CT where the tumors seemed to be melting away, as had been observed in the mouse models. But COVID came, and so we were stuck at home in those early days. And I started to think about this data set, and I realized that the PET-CTs that were being used to look for tumor metastasis are not specific for cancer. It's actually just a metabolic test that looks at glucose uptake and metabolic activity. And it turns out that a number of cardiovascular imaging folks have started to realize that these same signals are increased in patients with atherosclerosis. And there are now data showing that the aortic metabolic activity is increased in people with genetic forms of high cholesterol. And I think even more importantly, it's been suggested that this modality is sensitive enough to look at response to therapy. And there are studies even showing that you can tell which patients get randomized to low-dose Lipitor versus high-dose Lipitor and the quenching of that inflammation that can be observed. So we did a pilot study, and I think that this video didn't transfer. It's kind of a fun video, but it shows that the mouse aortas will become less inflamed once they're given this therapy. And so we hypothesized that the patients that were in that clinical trial for their cancer who had these tumors melting away might be having a simultaneous improvement in their vascular inflammation. Now, at this point, that first cancer company had been acquired by Gilead, and I called them and said, hey, wouldn't it be great if you let this academic head-in-the-clouds guy come and muck around in your clinical data sets and look at your imaging studies? Wouldn't that be fun? And they said, no, thank you, we're fine. You can go on your merry way. And so I realized that that's how drug development occurs. But I also realized that we had access to the de-identified scans at Stanford just through the faculty because those patients had been enrolled there. And so we went back and we were able to retrospectively analyze these scans. And we did this with the chief of nuclear medicine. And he got the scans and de-identified them and then looked at them in a randomized fashion. And what he showed was that you can see the metabolic activity in the brain, and then that's the thyroid in front, and then the arrow is pointing to the carotid bifurcation. And this is where plaque will build up in people before they have a stroke. And this was all just by pure luck because the patients with cancer in this study were old enough where they just happened to have atherosclerosis, even though they weren't enrolled based on that enrollment criteria. But what we found was after 8 weeks of therapy, there was no change in the metabolic signal in the brain or the thyroid, but the arrow shows you that there was almost complete quenching of the inflammation in the plaque. And regardless of how we quantified it, there was this improvement over time. And this was a stunning result. And so in my keeping with having lots of mentors look after me, I don't know if Lou is here today. I thought he might be. I've been fortunate to have also mentorship from one of John's dear friends, Lou Lang, who was chief of cardiology at Barnes-Jewish and then was the CEO of CV Therapeutics, brought the first anti-anginal drug to market in several decades. And I called Lou and I said, you won't believe what we found. And he says, okay, you've met John a few times, but what I'd like for you to do is come over this afternoon and let's show him the results. And so I happen to have this picture, Lily, and this was the first time, my laptop's there in front. I showed Lou and John those results from the human study, and John says, okay, let's start a company. We're gonna cure heart disease. And so that set us off on our merry way. We published the results, and we were able to set out, and we started a company called Bitterroot Bio now, and that's after the fishing river that I mentioned earlier with Irv, where we first hatched up this plan, trying to fish for rainbow and cutthroat trout on his ranch in Montana. And this was a wonderful time. And Lily will remember we'd get together, we'd come over and we'd brainstorm, and we had so much fun putting this group together. And we were able to leverage some really beautiful technology that was developed at Stanford that would more avidly bind CD47 and had some characteristics that we thought would enhance its safety profile. We were able to graft this onto a modified fusion protein with an IgG4 backbone that would have characteristics that we thought would reduce some of the hematological toxicities that have been observed. And we are now well underway. We have data showing that this has really potent effect in mouse models of atherosclerosis, much greater than what we've seen with the tool compounds that are out there. Last year, we completed our first in human phase 1 single ascending dose trial where the medicine seems to be fairly well tolerated. And although we're still working out some of the pharmacokinetics, we were able to launch the first cohort of our phase 2 study in actual patients with heart disease last year in Australia. And we're very excited to see how these studies will turn out. But as we are concluding where we are to date, I'm a pretty impatient person, and so we're trying to figure out and predict what we're likely to find if this trial does work out as planned. And so to do that, we've been partnering with people that run some of the largest biobanks in Europe. This is Gerard Pasterkamp from Utrecht. He and his team have the largest collection of atherosclerotic plaques from carotids and aortas in the world, and they've done very deep histological, morphometric, and genetic characterization of these plaques. And so we partnered with him to try to predict what we're likely to find. The first thing we did was we mapped the genetic profile of animals with dysfunctional efferocytosis onto the human plaques. And he has shown us that this will correlate with individuals who have the highest features of inflammation with lipomatous and fibroinflammatory plaques. These are the plaques that you don't want to have. He's also shown us that people who have dysregulated efferocytosis have the highest burden of inflamed macrophages in their plaques and are most likely to have clinical symptoms. They've also shown that you can characterize the vulnerability of the plaque, and that, again, a defect in efferocytosis correlates with the high likelihood of having a plaque that's likely to rupture, leading to that TIA or stroke. And then I think, what's really interesting is he can predict what's likely to happen if you were to restore the profile of efferocytosis, as we're trying to do. And he has now modeled that this will shift the plaques very commonly from a more to a less vulnerable phenotype, suggesting that we can expect some clinical benefit. Now, these are all in silico predictions, and so we're trying to validate this. We've now run through his AI algorithms, a random list of genes, and we were happy to see that this does not predict any change in the plaque phenotypes. He's shown that if you make a mouse more inflamed with endotoxin or LPS, that this will destabilize the plaque. And then, as predicted, if you give an animal a statin like Lipitor or Crestor, it will lead to benefit. But we were really happy to see that our pro-efferocytic therapies had the largest positive benefit. And I won't show it here, but we now have data that statins and pro-efferocytic therapies have orthogonal and synergistic effect. And we know that when we go into the clinic with standard of care, all of our patients will have to be on their lipid-lowering drugs, of course, and we may get not just an additive benefit, but one that's synergistic. So, as I wrap up here, I just think that it's been a wonderful journey trying to understand this defect in efferocytosis and explain the mechanisms thereunder, with the hope that this can now become a new translational therapy. I think that, as probably John would have thought about, when you're addressing these huge problems, you often have to do it in a novel and orthogonal way that goes beyond what the textbooks have written. And that's what we've tried to do here, leveraging unbiased human genetics. This has led us to this concept that inflammation resolution can be achieved, perhaps through these new immunomodulatory approaches. But at the end of the day, it's really about the joy of medicine and science, and I think that this journey with John has been so wonderful. I think about him often, and so in addition to thanking the group in the lab who did this work with us, I really wanted to, thank John and the memory. To this day, we still have our plaque up in John's memory at Bitterroot. He is somebody I think about often and I still draw a lot of inspiration from. And so, Lily, thank you for having me, and thank you all for your attention. I'll be happy to take any questions.
Questions? Yeah.
Very nice talk. I'm wondering if you found CD47 in that initial GWAS studies that you mentioned.
That's great. So no, it has not turned up, and that's been one of the things that has puzzled us. It turns out CD47 is very highly conserved and probably has a key developmental role. We now have some unpublished data where other factors in efferocytosis are showing up, and it certainly correlates with plaque vulnerability status. But you're right, it would have been great if we had seen it from a GWAS perspective. We think that some of these things are just so, there's so much evolutionary pressure on them. The gain and loss of functional alleles are almost non-existent, suggesting that pressure.
Nick, the statins are known to stabilize plaque. Were you surprised to find that they were synergistic with the antibody?
Yeah, I think that they work through separate mechanisms. They do have areas where they funnel together through some NF-kappa B signaling, but they branch back out. The fact that they will survive and cause this synergistic benefit is really appealing. Statins will actually calcify plaque. I don't know if any of you have had a coronary calcium scan. Once you have one and you get put on a medicine, patients want to have another one. I said, no, you can't do it because it's going to get worse, and we don't mind that. So I think of statins reducing the soft lipid-laden core, stabilizing, calcifying the plaque, and then the efferocytic machinery coming in to get all that inflammatory debris through the macrophages of the shoulder region. So we're really excited, and we hope that we'll get 1 + 1 = 3, not 1 + 1 = 2.
I have a couple of questions. Given the role of inflammation on heart disease, you know, with all the anti-inflammatories that are being used for IBD and all of the other diseases, autoimmune diseases, have people looked at the epidemiology of those patients on those anti-inflammatories and their heart disease?
Yeah.
And whether those are safe drugs or not for long-term treatment?
Yeah, they have. It's a great question. The NIH sponsored a study with methotrexate, which was negative, thinking that might be a benefit. But colchicine actually now has pretty robust data. So taking that old medicine for gout, and it will reduce inflammation and lead to reduced outcomes.
So like, how about the anti-IL-23
yeah.
and everything else?
Yes, yeah. So that's never been prospectively tested. Like, you know, if you have a lupus trial for TNF inhibitors or whatever, that's never been adjudicated formally, but they have done observational studies, and the observed to expected rate is much lower. So they do think that, you know, these powerful immunosuppressives will reduce myocardial infarction.
So my other question is you're trying to upregulate calreticulin, which is hard.
Yeah.
So you block. But can you actually think about doing it in a different way, is taking the calreticulin epitope and making a bispecific where you link that to an antibody FAB against a marker that's highly expressed on those apoptotic cells. So you get the level up very selectively, simply with a bispecific.
Yeah, stay tuned. We're working on that.
Okay. Good. I guess that means it's a good idea. What else? Other questions? Yeah.
Could the inflammation actually be caused by getting COVID? Oh, sorry. Could the inflammation actually be caused by getting COVID?
Yeah, I think we saw a big increase in cardiovascular events during COVID. A lot of them were thrombotic, but there were, of course, increases in myocardial infarction as well. Anything that's inflammatory will increase your risk. You know, we know that lupus patients, autoimmune disease patients have higher risk. This is why we're so excited to see some of these observational studies with, you know, TNF and IL-6 blockers. I think it's, you know, threading the needle. You know, I think Novartis has a large IL-6 trial underway in phase 3 for cardiovascular disease and people with chronic kidney disease. So we all expect that trial to be positive from an efficacy perspective. The question is, will it have a safety signal?
My last question is, how do you think about heart disease, because it's a chronic treatment, and like an anti-CD47, in cancer, you can think, okay, we can put up with the anemia and everything else. How do you think about these drugs, and when do you treat heart failure and heart, you know, disease? That seems like a harder drill than like IBD or something.
Yeah. No, it's a great question, and this is one of the real problems with how cardiology is as a discipline. You know, we wait till you've had 4 decades of atherosclerosis build up, then you have your heart attack, and then we go, you know, crazy. Repatha, you know, a PCSK9 inhibitor, they just had at last year's AHA a beautiful study giving it for primary prevention. They showed a mortality benefit. Yeah, so I mean, we probably need to be treating people sooner. I've been on Lipitor for over a decade now. I bet you I always talk about this in the laboratory, so people that work with me, I think that we should all be addressing these risk factors much sooner. And you're right, I mean, for the heavy hitters, for these things where do have side effects, you might have to wait till the disease is more advanced. But we're working on ways to avoid the hematological toxicities. You know, Tom's work with these nanoparticles that don't get into the spleen where erythrophagocytosis occurs have a lot of promise. And then our molecules, we're engineering them to avoid that.
Any other questions? Yeah.
How about diet? Mediterranean diet?
Good. I don't know. The question was about Mediterranean diet. Nutritional research is, you know, some of the hardest to have rigorously controlled, because often if you're willing to eat, you know, roots and berries, you're probably doing other things well, like not smoking and doing exercise. The Mediterranean diet probably has the strongest evidence for all of the randomized trials that have been conducted, and we don't know. Is it the olive oil? Is it the nuts? Is it the healthy fats? We're not really sure. Probably no harm in taking them, but the latest lipid guidelines got rid of fish oil by itself. We really don't know in a rigorous way if that definitely has benefit. And that's okay, because we've got new triglyceride-lowering drugs that are coming that will probably address that risk. So I would just say we don't know. Makes sense, but we really like to have hard evidence, and that's lacking because of all the confounders with behavioral science.
Any other questions? If not, join me in thanking Nick for a really terrific seminar. That was really great.
Thanks. Appreciate
Tough problem.
Awesome.
Now I'll have Ben introduce our next speaker, Stuart Schreiber. Ben?
Personal great pleasure to have an opportunity to introduce Stuart Schreiber. Stuart, along with our colleague and president, Peter Schultz, are generally and rightly regarded as the fathers of modern field of chemical biology. Their joint custody over this important discipline extends beyond its origins to include responsibility for many of the landmark achievements and the training of most of its current practitioners in both academia and industry. Well, it may be difficult for the younger scientists in the audience to fathom a time devoid of research at the interface of synthetic chemistry and cell biology. I can testify that this is largely how science operated before Stuart. This all changed in a radical way when his lab first demonstrated the power of organic synthesis to elucidate the mechanism of action of bioactive natural products such as the immunosuppressive agents, cyclosporin and FK506, which Stuart's group showed had the mind-boggling property of acting as molecular bridges to bring two proteins together, forming ternary complexes that altered signal transduction in the cell, a previously unheard of mechanism of action that Stuart coined molecular glues. So some of you may know I was not only a science major, but also a history major, and so I've always been kind of interested in the history of transformative scientific discoveries. And so I did a little bit of sleuthing, right? Because I remember when this science came out, I was actually a graduate student, and it felt like science fiction to me. It was just so incredible to read this work. And so over the years, I've gotten to know individuals who were in Stuart's lab at the time that these discoveries were made, and I've peppered them with questions, what was going on? How did you process this crazy biology and chemistry interface? And one of them, we chatted for quite some time, and he said that, they would have these lab meetings, and they were going over the complicated chemistry genetic interfaces that were trying to explain the mechanisms after the drugs. And then at one of these meetings, Stuart said, I think I understand what must be happening. It must be that these compounds bind a protein, let's call it a presenter protein, and that doesn't do anything on its own. But then that binary complex finds another protein in the cell, and then that neither the small molecule or the original presenter protein bind on their own. And then they form this ternary complex, and that's where the biology comes from. And this former student said that the lab said, oh, that's a really interesting idea. Yeah, it makes a lot of sense. And then Stuart went back to his office, and they all looked at each other, and they said, he's lost it. What are we gonna do? Our advisor's completely gone crazy. So it just shows you, right, that outside-the-box thinking requires an open mind, right? That even the youngest scientists, who should be the most open to new ideas, had trouble fathoming at that moment. So since that time, Stuart's discoveries opened eyes to the remarkable activities that small molecules can display in cells. He really founded the field of what's now referred to as induced proximity, which has not only impacted fundamental science, but medicine in incredible ways. It's the basis of the mechanism of action for breakthrough drugs such as the immunomodulatory agent rapamycin, the multiple myeloma therapies called the IMiDs, and most recently, the Revolution Medicines anti-RAS drugs, which many of you probably heard about this week as they reported phase 3 data in pancreatic cancer, really revolutionizing the opportunity to treat that devastating disease. So I have one ask of you in the audience, that when you hear about these transformative medicines, please keep in mind the long and arduous journey that it took to develop them over multiple decades, and the often innovative academic origins of these concepts and medicines, in this case attributable to the field-launching research of Stuart. I'll finish with just some personal thoughts. I've had the very good fortune of being able to spend time with Stuart in a variety of professional and social settings over the years, and I can say In each of these instances, I'm a better person as a result of these interactions. Sometimes I gain scientific knowledge, like how can one interface innovative chemistry with human biology to solve challenging problems, but without question, the most valuable insights have come from understanding what it means to be a generous and graceful mentor, colleague, and friend. So Stuart, it's always wonderful when you can take the time out of your busy schedule to visit us here at Scripps. We look forward to your lecture.
Ben, thank you very much. That's actually really helpful, and I'll try to build off of some of the things that Ben said. Good afternoon, everyone. It's just a great pleasure to be here. As Ben mentioned, I've had the good fortune to be here a number of times and smart enough to actually extend those stays. I took some of these pictures. When I stay, I explore your great neighborhoods, and I've actually, visited and stayed and taken an apartment out in the various neighborhoods. I hope you recognize some of these. So I kind of feel that Scripps is a, home away from home, a second home. It's been an opportunity in coming to Scripps to develop fruitful collaborations and scientific exchange. But far more importantly is that it has been an opportunity to develop lasting friendships with very special people, some of whom are shown and some of whom are actually in this audience today. But we're here today for a very special reason, as we've heard, to honor John Martin. I think it was great learning, many of us did, from the introductory comments from Pete, and then from people who knew him well, like Nick. I actually did not know John personally, but our careers sort of tracked in parallel, and I certainly knew of him. Part of that was due to the fact that the launching of Gilead and a company I'm familiar with, Vertex Pharmaceuticals, occurred at the same time. Gilead here, of course, and Vertex, although in Boston, also has its San Diego origins. My co-founder of Vertex is Kevin Kinsella of La Jolla. And in fact, if any of you ever have a chance to visit this, if you can see my pointer, that's the Kinsella Library and Museum in La Jolla. And it's open to the public. It's really quite amazing. So I didn't know John personally, but I tracked his work, and for reasons that I hope will become apparent. So you may be, Gilead, of course, we know, developed a franchise in the anti-infective area and had transformational impact on that, much of which is due to John. You may be surprised that Vertex actually also started in the anti-infective area. We were working on the HIV protease, and we're really committed to making drugs for the treatment of AIDS. We did not succeed, certainly to the level of Gilead, and the reason is very obvious, that we did not have John Martin leading our efforts there. Now, I put up these structures to make another little personal connection. Many of you all know how hard it is to make a drug. It's even harder to make a drug that has two components, and almost unheard of to make one with three chemicals in one pill. I'm actually only aware of these two. One is a tripla, one of, I don't know how many drugs John was responsible for developing. It's in the mid to high teens, if not more. This is one. And it shows, Pete introduced him as a scientist, a businessman, and a humanist. I agree with that statement, because in this case, as difficult as it is to make a drug with three components, partly, for example, you can imagine you try to optimize the dose, but now you have to do it with three different components, and ultimately it's fixed by the stoichiometry of the pill. But in this case, as I understand a tripla, not all of the chemicals came from Gilead. And so he had to cajole colleagues in the pharmaceutical industry to come together and do the right thing, because I always viewed John as someone who felt it was a moral imperative to make drugs, and he really, really meant it. Okay, I'm going to use that as a segue to start telling you what I want to describe today. So Vertex has made a drug also with 3 components. I'll tell you a little bit about this later. It's called Trikafta. Vertex adjusted from the anti-infective area to take on a human biology-driven approach to drug discovery, as we heard Nick describe. And so they focused on cystic fibrosis and the target of CFTR, a protein that, when mutated, causes this disease. And so their franchise drug right now is Trikafta. It's an amazing medicine, and it comprises these 3 chemicals. They have this amazing property. They bind the same protein, the CFTR protein. And so in the Vertex case, we were able to overcome this challenge by the remarkable property of cooperativity. And I'm going to be talking today about cooperativity. And here's an example. If you tried to optimize the dosage of the 3 individually and then put it together, you'd miss the main point. And the point is that these chemicals bind the same protein, but they do so with high 3-way cooperativity. Every binding event increases the affinity of the other. And so they could use very low concentration of these and make this drug. All right, so I do want to tell you about the origins of my lab's efforts, has been mentioned, in proximity-inducing drugs. And they fall into these 2 categories. One that has this property of cooperativity, we call those molecular glues. And the others we call bifunctional compounds, because they largely bind 2 proteins independently, and we link them up with a linker. So they lose that cooperativity, but we still get the proximal relationship, which is key. So to calibrate you, when the initial discoveries were made, it was the late 1980s, and at that point in time, a major problem in biology was the mystery of signal transduction. How is it that an extracellular protein can bind a surface receptor, never enter into the cell, and yet influence all of these intracellular events all the way into the nucleus, with specific genes being turned on and off? And so in the late 1980s, the details were unknown, but it was becoming clear that protein-induced proximity was playing an outsized role. So for example, the extracellular growth factor could bind a receptor with 2-to-1 or 2-to-2 stoichiometry. Many of these have enzymatic activity in their tails. They're brought in close proximity, and you increase the rate of transphosphorylation without ever entering into the cell. This was very satisfying from a chemical point of view, because we chemists are very familiar with the concept of high effective molarity, or effectively intramolecularity, increasing the rate of chemistry. And this goes all the way into the nucleus, all the way into regulation of specific genes, as maybe I'll try to touch on a little bit later. So it was with this chance discovery, studying 2 natural products, that small molecules can do exactly the same thing, that you can imagine how incredibly excited we were, because, whoa, we now have a way with chemistry to target inducing protein-protein interactions, this key to information transfer in biological systems. So I'm going to tell you a little bit about what one can do today, some new examples of increasing, decreasing lifetime of targets, increasing, decreasing activity of targets, changing cellular localization, and many others. So the example that Ben alluded to here is these 2 natural products, cyclosporin and FK506. They have very different structures, and yet Jun Liu, who was a postdoc in the lab at the time, using essentially affinity chromatography, led to this realization that these 2 compounds are targeting the same protein phosphatase named calcineurin. But the unusual thing, and there's a couple of unusual things. One, they're not binding in the enzyme active site. That's where the asterisk is here, here, and here. So they're binding to an exosite, and they actually block the entry portal for some substrates, but not all of them. So another little twist. We had substrate selective inhibition. And then lastly, the compounds don't bind calcineurin. We use these compounds to discover we call them presenter proteins, cyclophilin for cyclosporin and FKBP12. We use actually FK506 and rapamycin to discover FKBP12. And those presenter proteins don't bind calcineurin. It is only when you put them together that they act through cooperativity to get high affinity and high specificity interactions. Now, in the title here of the slide, I use the term inflection point. Now, that's a term that I learned from the tech world and tech investors. They are always looking for what they call inflection points. And a poster child example in the tech world of an inflection point was an engineer at Apple decided to add a GPS chip to the iPhone 4S to enable the new app, Find My iPhone. And I'd like to think there's an analogy here because that worked, and that app works. Okay, but what happened was people working in the hoteling industry dropped what they were doing and realized all these people are walking around with GPS chips in their pocket, and they created Airbnb. And people in the ride-sharing business who were trying to figure out how to link cabs with people said, we don't have to do that anymore, and they created Uber and Lyft. We knew that rapamycin was a third molecular glue, but we were off with Jerry Crabtree using cyclosporin FK506 to study what we now call the calcium-calcineurin NFHE signaling pathway. So we came back to this when Eric Brown, a graduate student, joined the lab, used basically the same approach that Jun had done with calcineurin, and discovered that indeed rapamycin is a glue. It also uses the same presenter protein, but it targets what we called the mechanistic target of rapamycin, because the target of rapamycin is FKBP12. And David Sabatini, when he was a graduate student in Saul Snyder's lab, made the same observation. We published this at the same time, and David has gone off to do amazing work in understanding the mTOR-mediated nutrient response network. Now, at that time, we thought, we only had these three examples, and we thought they're very complicated structures, and they're all the products of 1 billion years of natural selection. Maybe this is just something very rare and not accessible to us chemists. So we had two different, I'll show you two different ways to think about this. So one was another serendipitous discovery by Steve Haggerty, then a graduate student in my lab. And Steve discovered a very simple, off-the-shelf synthetic compound, one-step synthesis, low molecular weight, that was able to take dynamically interacting proteins and clamp them together. Similar principle, but now not proteins that never interact, but weakly interact. And we call that synthetic stabilizer A. But that's exactly what the very complex natural product, Taxol, and several other natural products now, is able to do. But you don't need natural selection. You don't need complicated structures. I'm beginning to think now that even it may be an advantage to have especially low molecular weight compounds as molecular glues. I put this in for Ben because we've had lots of fun discussions about presenter proteins and basically the crystal structures of the rapamycin, and then the compound on the right, which really emphasizes this point of synthetic compounds. This is a compound that was reported by Felix Hausch recently. They made a library of 1,000 compounds, and they found a rapamycin out of it, a completely synthetic compound. And crystal structures now compare these, show that same target, same hotspots, but the presenter protein is rotated by 90 degrees, that's FKBP12, and the flexible loops around the hotspot are engaging the proteins in completely different ways. This is a theme that we've now seen over and over, as I'll try to get to. Here's another one, another natural product recently found. It binds a coiled coil of another protein, CP250, which I think would be very difficult to do with a small molecule alone, and yet using this presenter, which now uses those flexible loops in a completely different way to engage with high affinity, high specificity, a coiled coil. So taking a step back, and again, lots of great discussions with Ben about this, we've been beginning to think that chemicals binding proteins in cells, I think many, at least in the past, would equate these with inhibitors as a synonym, you know. And in fact, when small molecules bind dynamic proteins, they change their biophysical properties. We can think of the small molecule protein as a neoprotein, and the altered biophysical features often change the interactome. I'm beginning to believe more often than not that these are not rare, we just weren't looking for them, and they're everywhere when compounds bind proteins. Now I want to, it says, two things I hope you walk away with today are these two analogies. Molecular glues behave, one, like post-translational modifications that I mentioned before. And two, they are also like missense mutations that have been enshrined by natural selection. And what I mean by that is that mutations presumably occur largely randomly throughout the genome, but the ones that change amino acid identity and that we know about are the ones that we know about them because of the process of natural selection. They've been selected to do something. Now, we learned from Jim Wells a number of years ago that when molecules bind proteins, they don't do so randomly. They bind in what he called hotspots. These are evolutionarily conserved regions of proteins that are normally involved in protein-protein interactions. So, it turns out missense mutations, amino acid changes that we know about, they don't occur randomly either. They occur in or around hotspots. So, what do they do? Well, in many cases, missense mutations function by changing the interactome. They induce neoprotein interactions. So this, again, is very satisfying from a chemist's point of view. You have a hotspot, and you change an amino acid side chain, you recruit a new protein. It's not unlike the wild-type hotspot having a small molecule mimicking an amino acid side chain, inducing a protein-protein interaction. A gazillion examples of these now that happen, say, like every week or every month. This is an older example, but I really like this for a number of reasons. This is work out of Novartis. They were interested in the MALT1 protein. They chance discovery of a binder, crystal structure of it. The first thing they noticed is that the compound displaces the indole ring of tryptophan 580. The triaza-indole sits exactly where the indole is when the tryptophan is functioning as a intramolecular glue to keep the two domains of the protein together. This gave them the idea, knowing that there's a Mendelian disorder associated with the mutation of that tryptophan to a serine, and it's an unstable protein, unable to signal to NF-kappa B, that maybe they could rescue this with a corrector, a glue corrector. And that's exactly what this compound does. It regains stability, it glues the two domains together, and I call this a molecular prosthetic. And then the analogy I've already made to post-translational modifications, you know, when those tyrosine kinase activities phosphorylate, well, now there's proteins inside the cell that have SH2 domains that bind phosphotyrosines. And that includes lipid kinases that are now recruited to their substrate, the lipids in the membrane. And on, all the way into the nucleus where, you know, acetyl groups emerge on the lysine tails of histone proteins, where nucleosome remodeling enzymes bind the acetyl lysine and are now in close proximity to the nucleosome, allowing the transcriptional regulation to occur only at that locus in the genome. The IMiDs were mentioned earlier, lenalidomide and thalidomide. I have an interesting story I want to share with you. First, it was Handa, 16 years ago, who discovered that the target of these compounds is cereblon, a component of an E3 ligase. 5 groups working together in collaboration in 2 pairs. My lab working with Steve Carr and Ben Ebert, and Jay Bradner and Bill Kalin together discovered that these are molecular glues. They induce neoprotein interactions, and since cereblon is part of an enzymatic complex with E3 ligase activity, increases the rate of ubiquitination, and then proteasome-dependent degradation. Well, recently, Christina Wu discovered, studying an old post-translational modification, old meaning we've known about it for 40 years, but really didn't understand what it was doing, And notice that some proteins will undergo this PTM where asparagine or glutamine side chain will cleave the main chain, leaving on the C-terminus now an imide. And indeed, that imide, like thalidomide and lenalidomide, binds cereblon, which then ubiquitinates and degrades the protein. So it's part of the protein degradation pathway. This working in cis, but the glues working in trans by bringing in neoproteins. And then Ben mentioned, again, in medicine today, it's hard to miss the fact that there's just this notion has been very extensible, as we had hoped in the very beginning, since it seems so fundamental to information transfer. So this is the revolution in medicine. One of their RAS-on inhibitors, they took cyclophilin, that now we know multiple natural products that bind cyclophilin. By the way, they're all glues to different proteins. And equipped it with an electrophilic group to engage the nucleophilic G12C oncogenic allele of KRAS, and then structure-guided design to optimize the protein-protein and small molecule protein interaction. That is the basis of that compound. It's and that's why I justify calling this a GPS inflection point. And the last example, but again, there's just many, is what I said in my introduction. This is Trikafta. So here we have it. This is CFTR. This is a Mendelian disorder. There are multiple missense mutations, but a primary deletion mutation where phenylalanine 508 is skipped. The protein is unstable. And what Vertex actually originally, Agaron, is that right, Agaron? Aurora, sorry. Aurora, here out of San Diego, discovered binders to CFTR. They found 3 different hotspots. By the way, there are others. And now we know they bind, as I said, cooperatively. So they reinforce the interaction of each other. And what do? Well, they contort the protein in a way that takes it back to the native structure. And they confer on the mutant form all of the wild type properties, these correctors or prosthetics or what. I don't know if Gabriel Lander's here today or any of his group member, but there's a beautiful study from the Scripps here showing, actually bringing a whole bunch of things I just told you about together in one study. So one, intra and intermolecular glues. So these imides bind an open conformation of cereblon and first act as an intramolecular glue to a closed conformation. If you get that far, they're candidates to recruit intermolecularly neosubstrates. Even more recently, I don't know if you had a chance to read this, but it's kind of remarkable in the light of what I just told you about CFTR. So the Lander lab, working with Benovitz and Wu, have just reported the discovery of another hotspot on cereblon and a chemical that binds there. This is sounding a lot like CFTR, because what does it do when it binds there? It increases the affinity of the interaction of the compounds at the first site. So again, you have this cooperative way to start all kinds of ways of thinking about new medicines now based on cooperativity. All right, so with Jerry Crabtree studying signal transduction, we got excited about how do we not have to serendipitously find one glue after another, but maybe find a binder to a protein, find a binder to another one, link it up with a linker, and make these bifunctional compounds. We would forego, unless we just got lucky, cooperativity, but we would remain the intramolecularity, the high effective molarity of the enzyme and its substrate, whereas they not normally interact. So a 10-year period of collaborating with the Crabtree lab, we discovered many examples of this. So one of the very first is shown here. So I like to say that Peter Howley is the originator of the targeted protein degradation field. And not surprisingly, based on what I told you, he discovered it through a protein inducer of protein-protein interactions. So he studied the oncogenesis of the papillomavirus and cervical cancer and discovered that a viral protein, E6, is a protein glue. And it brings together two host proteins, an E3 ligase and P53, ubiquitination and degradation. So Peter is a good friend and also a good friend of Jerry Crabtree, so we worked with him, and we made fusion proteins of E6 with an FKBP, and all kinds of target proteins with an FKBP. And then we made a compound we call FK1012 from an olefin metathesis of FK506. So it's no longer a calcineurin binder, but it binds two FKBP12s. And indeed, these compounds induce protein degradation. Much to my chagrin, a pharma executive at Ciba-Geigy at the time stole Martin out of my lab. I tried to get him to stay because I thought this is a really promising finding. But luckily, Martin's lab mate was Craig Cruz. And, you know, Craig has, in his independent career, together with Ray Deshay here locally in Amgen, have done a beautiful job in showing how, you know, these PROTAC molecules can do the similar thing in a wide range of substrates. The first example we used of showing spatial and temporal control in animal wasn't with targeted protein degradation, but cell degradation. So we made a transgenic animal, restricting the expression of a transgene with the FAS apoptotic signaling domain, a proto-caspase, fused to an FKBP, and a myrcellation sequence. We restricted the expression using the LCK promoter, made this animal, it made the transgene, but it was a completely unremarkable animal. Nothing we could see different about it other than making a protein. Until we treated that animal with FK1012, now you get to see the structure. A postdoc, Steve Diver, made in one step this bifunctional by the olefin metathesis reaction. And when we treated these animals with FK1012, we could induce the interaction of the proto-caspase domains. They're weak proteases. They cleave each other and activate the death signaling pathway. And this worked in humans. Jerry and I started a company called Area Gene Therapeutics, and a postdoc in Jerry's lab named David Spencer started a company called Bellicom. At Area Gene Therapeutics, we made this synthetic version of FK1012, and in this collaboration, leukemia patients were given a bone marrow transplantation, but prior to, after extraction of hematopoietic stem cells, they transduced them with the exact same construct I just described, myrcellated FKBP12 with a FAS domain. Why did they do that? Well, 5 of the leukemia patients treated with bone marrow transplantation developed graft versus host disease. And in this New England Journal of Medicine paper now, 15 years ago, a single dose of Remeducid eliminated the graft versus host disease because it reached the grafted immune cells and induced the apoptotic pathway. Jerry's lab in particular has done a great job of expanding this in so many ways of controlling biological processes. And Area Gene Therapeutics made kits available to academic labs, 1,200 of these given away and over 2,000 papers published, all kinds of extensions. So I wanted to show you some things that we've been doing in my lab more recently. I'll give you 2 quick examples, both coming from a young physician scientist in my lab, Will Gibson, who worked jointly with Matt Meyerson. Will is an oncologist. He's now on the faculty of Harvard Medical School and Dana-Farber. He was interested in the big important challenge of targeting P53 mutant cancer cells. And so he came up with this idea. I'm sorry, I'm a little ahead of myself, but all of this was stimulated around P53. So one was transcription factors more generally, and the class of transcription factors that have nuclear export sequences. These are transcription factors that reside in the cytoplasm until they receive a signal to get into the nucleus. And so he thought, well, if I use a bifunctional compound, I could bring it together with a protein with a very strong nuclear localization sequence. And the little video is showing what happens when you take this cytoplasmic protein, treat with this bifunctional compound. And then picked it judiciously so that the same protein had a very strong transcriptional activation domain or activation properties. And this was an effort, a successful effort to rewire cell circuitry. Adding this compound turns on the otherwise silent IRF1 transcriptional pathway because he targeted IRF1, normally cytoplasmically anchored. And now, more specifically to P53 is this compound. This also has some San Diego origins. This is a compound, P5301, that Will has made, where we start with a binder made by PMV Pharma, and that's Rich Hyman's company, Rich, the executive chair, and Arnie Levine, his partner. It binds a particular missense allele of P53. And it's linked now to a general cytotoxin. No one would think about using this compound in cancer because it's very good at killing cells indiscriminately. And he hoped that this compound would be a way to target P53 mutant cells. So how on earth would that work? Well, first over here from the DEP map, this just is a reminder that there's been many, many papers, many efforts to find synthetic lethal targets for mutant P53. If the cancer's mutant, maybe now there's a protein normally not dependent and it's now dependent, and that has failed entirely. No one has found successfully, I should say, a dependency. On the other hand, when Will looked at the Cancer Cell Line Encyclopedia, at the proteomic data, and asked, of all of the genes, and they're encoded proteins, are there any proteins that are upregulated when P53 is mutant? And the answer, as you can see, is very striking. It's one. It's massively upregulated. It's P53 itself. So in a funny way, I think we can say the synthetic lethal of mutant P53 is P53. Now, what does that mean and how do we exploit that? Oh, by the way, I said Will is a clinical oncologist, and his surgical friends in oncology know this very well without the CCLE. So surgeons wanting to get a good margin around a P53 mutant cancer will histologically stain for P53. And what you see here are different tumor samples where wherever the cells are dark, those are the cancer cells, and the stroma and the tumor microenvironment are not lighting up, even though they're making P53, just 100-fold less. And what is this? How does this work in oncogenesis? Well, first, the missense mutants are dominant. P53 is a tetramer. It blocks the binding of the tetramer to enhancers, including of that found in the P53 degrader, MDM2, another E3 ligase. And so this mutation fails to have P53 in an autoregulatory loop turn on its degrader. So what happens? You get massive upregulation of P53. And so the notion was then there would be a sink for such compounds, and we would selectively kill P53 mutations. This is a much longer topic. Is it really a sink effect? Is there something else going on? But in the paper that was published last year that's cited at the bottom, there's evidence that this unique compound is very selective for the mutant allele of P53 that the compound binds and not others, and that other compounds claim to be P53 dependent killers showed no activity in a similar assay. Okay, this takes me in the last couple of minutes to this beautiful collaboration with Ben's lab here at Scripps. Ben's group and my group had sort of independently merged on this idea of a binder-first approach. Like, chemicals are much more than inhibitors. We should find binders, and then they're going to confer some new activity. We just have to find out what that activity is. So on Ben's side, he brought this powerful method of discovering binders, even better in intact cells, chemoproteomic approach. And so what we tried to bring was something complementary. One was some chemistry the notion that hotspots are three-dimensional topographies, and so high sp3-rich compounds may be particularly good as functioning as binders, and even stereogenic elements might be useful, especially for structure-activity relationships. So we got, about 10 years ago, we started meeting monthly, sharing all of our ideas and so forth, and it was a fabulous time. Bruno Malio was a postdoc in my lab, and actually, I don't know if Pete's still here, it was Pete who said to me on one of my visits, Stuart, why don't you just embed yourself here at Scripps? And that's what led Bruno, fearless Bruno, to come to Scripps and be the embedded joint member. And so I hope you all will get to know Bruno, who's a spectacular scientist and thriving here at Scripps. By the way, the compound shown that came from this chemistry we call diversity-oriented synthesis, and that's a compound that Bruno has championed, which is now in phase 1 clinical trials as a novel anti-malaria agent. We thought, okay, in addition to chemistry, we would bring a screening approach that we hope would be complementary to the chemoproteomics. So we turned now to another Scripps influence of Richard Lerner, Sidney Brenner, their invention of DNA-encoded libraries. And so what we thought we would do is, instead of just making libraries with a single core and a lot of appendage diversification, we would make the cores themselves and the stereoisomers of them diversification elements. And we built a 3 million compound DNA-encoded library led by Liam Hudson. This just shows you that here's a 3-1-1 bridged system, a Spiro 5-5, a Spiro 4-5, a fused 6-7 ring system, and that fused 4-8 that I showed in the previous slide. So we still have appendage diversification, but now we're moving those appendages off in lots of different directions by the very different core features. Very first time we used this library, a binder was found of a protein of interest to Xuan Lu, and sure enough, it's a molecular clamp. She was interested in an oncogenic allele of IDH1, which is in equilibrium with its monomer and dimer, and showed by isothermal calorimetry and differential scanning fluorimetry that it was gluing them together. We would say clamping them together, just a little side. And I'll finish with the last thing we tried to bring was some other assays that might be useful for discovering chemical inducers of proximity or molecular glues or bifunctional. So at the bottom is the Richard Lerner concept. You take DNA-encoded libraries, incubate with a protein of interest attached to a magnetic bead, and then sequence the barcode that tells you the chemical history of the compound. Now, these are noisy experiments, and you got a lot of DNA around that will non-specifically associate with magnetic beads. So Lerner taught us to sequence the barcodes with the protein and without. And he called that ratio binary enrichment, and one looks for the greatest binary enrichment. First thing we did, this is work of Jeremy Mason, a postdoc who joined my lab trained with Bill Rausch. He said, well, why don't we look at the ternary enrichment by pre-selecting a target and a presenter. And now what we do is compare the barcode enrichment of this condition to this condition, and we call that the ternary enrichment. And that has worked over and over. It's a very robust system. In Jeremy's time, what we published here was the whole family of BRD proteins, and every single one we found bifunctional compounds that would induce association. We picked an E3 ligase, the E3 ligase, VHL ligase shown here, so that we would have a functional readout. These are indeed PROTAC compounds, but they're discovered in one fell swoop, not a binder, then a linker. All three comes right out of the screening deck. And in some of the papers that are in press or been published or and submitted now, very robust systems. Look, it's transcended to lots of other examples. So last thing is, Xuan Lu, who I mentioned earlier, she came in and said, why don't we do something that's just data analysis distinction? So why don't we analyze our data by looking at the ratio of the ternary enrichment over the binary enrichment? Could this be a way to screen for cooperativity? Because the barcodes now are going to be compared to this situation. So the ones where the presenter is enabling greater affinity could be the cooperative ones. And that has worked, again, over and over. Recently, Jurian Tan, a joint graduate student with Ramnik Xavier, said, how far can we take this FKBP presenter? Well, it turns out you can take it far. Here's a metabolic enzyme that's cooperatively glued together with a compound right out of the screening deck that takes FKBP12, and by the way, from this crystal structure of those flexible loops, they're finding yet another way to interrogate into the neighboring protein. I think I've run out of time here. Just very, very briefly, Ben and I have started a company together with our colleague, Michael Rosbach, based on these glue concepts. And this is not our results. This is a result of that company called Magnet Biomedicine. But I think it's yet another little twist on how this system can be used in drug discovery. Magnet has been trying to take advantage of the cooperativity here by making drugs that are otherwise inactive, but only work with the unique proteome of the diseased tissue. This is an example that the CEO, Brian Safina, gave me permission to present here. This is a molecular glue discovered by this Dell screening, and it's targeting, in this case, the tissue is the vasculature, the extracellular vasculature, where there's all kinds of unique proteins, the proteome of the vasculature. And now you can start to think about small molecules hitting cytokines and cytokine receptors, as is shown here, the way antibodies do. That is the hope of Magnet. So, look, went over a few minutes, forgive me. The key messages here on my acknowledgement slide is, especially for the young generation, science is becoming more interdependent. You're going to find tons of opportunities to collaborate. And the key message is choose your collaborators carefully. Yes, you need to have the right skill set to join together, but you want high integrity people, and even better is if you get lucky, as I have in the case of Jerry Crabtree, Ramnik Xavier, and Ben Cravat. They're just really decent, fun people that you really love talking science with. And so my appreciation to them and the group members who I've listed on this slide. Thank you.
Thanks, Stuart. That was fantastic. Time for some questions. Yes.
I have no science background. I don't know any of this stuff that you're talking about. I saw some of the stuff, and my mother had myasthenia gravis. And I'm thinking what you talked about in the first few slides seems very interesting, what you're talking about, because of myasthenia gravis, the lack of the chemical to induce muscle movement of the interior muscles. I was wondering how this factor, how what you're doing factors in to solving some of that MSMD, MG.
Yeah, I'm not doing anything in that area. I'm sorry to say, and sorry for your mother's challenges. But so I'd want to sit down more seriously with you to really figure out how to do this. But I can imagine all kinds of ways. I mean, I'm really excited about the idea of rewiring transcriptional circuitry and find, what is the transcriptional circuitry in the healthy state as opposed to the disease state? And can we glue together a transcriptional regulator to the right loci in the genome and turn on that whole program to take that disease state back to a healthy state? But there are many other approaches that, I'd want to work through with the experts before I pick one.
Well, thank you very much for what you're doing.
My pleasure.
Yes. Fantastic talk. I was curious, so a lot of these initial compounds you discovered are natural products, and I was curious, do they act as molecular glues in their native environments? Because these things weren't evolved to treat our cancer. They were
Correct.
thing. So do you know if they're molecular glues in that context as well?
I think that's 100%. There's no doubt this is how they're functioning. And so I have a simple-minded view, and it's best supported with probably cyclosporin and FK506. So these are So let's take FK506. It's made by a bacterium. It's very common. You go out in your garden, you'll find one's making FK506, one's making rapamycin. So what are they doing? Why are they doing this? Turns out cyclosporin and FK506 are very good antifungal agents. Rapamycin originally was discovered as an antifungal agent. And so, this is just microbial warfare. And, a spore is floating along and it finds that same orange peel that the bacteria was munching on. And so the bacteria are making this knowing that the fungi are making FKBP12. It's brilliant, right? It's that tissue-specific drug action. You make it knowing that there's a contingency for it being active, and you're protected because you don't have the means for cooperative interactions. I think that's one explanation, kind of microbial warfare.
Yeah, I love this. This opens up organic chemistry, to all of these new targets. So we're not strictly focused on biologics these days, which pharma has gone to the extreme recently. But I have a question on the bifunctional molecules that are reversible. I don't actually understand how the energetics work, because if the proteins don't interact, you're binding one molecule, there's an on and off rate.
Correct.
On that side, there's an on and off rate with the other protein.
That's why not all bifunctionals who we're finding are working in the clinic, for example. Yeah. A lot of the PROTACs, it's been hit or miss. And I think it's a lot of the kinetics, and I wish Jeff Kelly were here. He calls this, he has a term, I actually cited it on one of the slides, both chemical and genetic. He focuses on the kinetics of the interactions. The glues, I originally named these compounds glues because I was working with Don Wiley when he had just discovered the extra electron density in MHC proteins. He figured out it was the antigenic peptide, and different MHCs binding different peptides would recruit different T cell receptors. And it seems so much like this. But the amazing feature of the MHC system is the kinetics. Now, they're actually weak binders, but the off rate is on the order of weeks. And these glues, the glues, cooperative glues, same thing. Very long residence time. Not true of many of the bifunctionals. Yeah, so the bifunctionals are easier, but I think most people are today getting away from them as much as possible in drug discovery. They're easier conceptually, find a binder, link them up, but cooperativity is really powerful, and altering the kinetics of on and off rates is maybe a missed opportunity, but.
Yes, I have a follow-up question on that, actually. You made the poignant comment that small molecules can be so much more than antagonists. They can traverse from agonist, antagonist, to neoagonist. And then I was thinking that, especially with your DEL screening now, could the DEL screening be quantitative enough that it could pick out gradations of the stability of a ternary complex, such that one can imagine a small molecule that binds tightly to a presenter and then just touches the protein target lightly, so it can do some, let's say for the sake of the argument, the presenter is an enzyme, so it does some chemistry on it, then let's go, right? Yeah. So it becomes like an agonistic element, and then of course as you look for the compounds that are even more cooperative in the DEL screen, they become antagonists because they trap.
They trap it,
right?
Essentially irreversibly. Yeah.
Is that theoretic?
I think theoretically it is. I think what I can say is that the range of the presenter ratio, we call it, the ternary enrichment over binary enrichment, is quite significant. And we did spend a fair amount of time looking at what does it mean to be a lower presenter ratio and a higher presenter ratio. And I don't know that there's a simple answer to your, but I think intuitively buried in there is going to be, with more experience, a way to look at the screening data itself, just to give you some hints of these features other than simple cooperativity.
Other questions? Okay, if not, let's thank Stuart again, and then Pete, do you want to have any final words? You're coming down this. Okay, good. Oh, that's right. Yes, thank you. Okay, great. Excellent. Thanks, Stuart.
Hi, it's my pleasure to introduce Greg Alton, the final speaker today. Greg actually, I think, spent 20 years at Gilead with John. He was the chief patient officer, and as such was responsible for the interactions with the government, with the public, and actually facilitating John's vision of providing low-cost access of drugs to low and middle-income countries. Greg was also the general counsel at Gilead, the compliance officer, I think a job nobody would wish on anybody, and has been actually really helpful to us in thinking about how we can impact with our global health efforts hepatitis and other diseases around the world. I noted Greg got his degree from Berkeley in 1989. Lately, I'm meeting people who took organic chemistry from me, okay, at Berkeley. But I guess if he did, he probably wouldn't be here today, okay? And then got his law degree at Stanford, so he's a well-trained chemist who's also a lawyer. So Greg's gonna say some words about John and the time at Gilead. So Greg, I'll turn it over to you.
So I think it's time for me to change that photo. That is, I think that was taken right after a triplet was approved, if I recall. That was like 2010 or so. So I'm going to do a couple things. There's three things I want to do today. First of all, I want to talk about some lessons that John taught us that I can maybe pass on to you. I worked with John for 20 years, which, by the way, makes me the junior person in this room of people who worked with John, because the other people in this room worked with John even before he came to Gilead. So I think, Bill, you followed him from Syntax, as did Swami. You came from Bristol Myers with him, and Tyann, you came from Syntax with him. The other thing I want to say before I start, I'm not a scientist, although I found out I'm not the only non-scientist in the room. There may be others, I don't know. So bear with me on that. So I want to teach you some of the lessons John learned. I do want to talk about John, what he did with what we call our access program, John's commitment to making our products available globally, and what that really meant to him. And finally, hopefully you can learn a little about who John was as a person. He said that John was a strong, he had the rare characteristics of a strong businessman, humanitarian, and scientist. Absolutely true. Very, very rare to get all three of those things in one person. But I think as you see, when you have people who work with John for 20, 30 years, he also was just a really good friend. John just had a really way of meeting people, working with people, getting to know people, and really valued personal relationships. He was a very personal person, a very down-to-earth, kind of a normal guy. John had a big ego, don't get me wrong, but he didn't let that ego get in his way. He didn't like carry that on his shoulders. So that was just a little bit about John. So I'm going to talk about these lessons about addressing patient needs with breakthrough science and innovative access, and hopefully through that you'll get a sense of who John was. So when I think about John, I think about sort of two things that how he approached things. He had a scientific contract and a social contract. On the scientific contract, I think a lot of people talked about this, but innovate towards what patients need. I think it was said, in Stuart, in your presentation, you talked about the antisense technology. I think John recognized that antisense, although great science and has actually proved to be quite good, at the time wasn't ready for prime time. He wanted something that was going to address patients sooner. Great scientific progress is doing something others said was too hard, couldn't be done. And if you think about what John did with the nucleotides, Swami, you can attest to this. BMS dropped it. Other companies just thought it was just too risky, too hard to do. John said, no, this is going to be the game changer. John was also really big on intellectual property and access to capital, and he was very strong on intellectual property rights, how that was really a basis for access to capital and what allowed science to develop. So he was really strong there. But at the same time, John had the strong social contract. I think that was really unique about John. He really wanted to pursue sustainable solutions, looking across healthcare for infrastructure, diagnostics, how he can use that science and really accomplish the goal of what we really want to do, which is treat patients, make patients live longer, live better, and prevent disease. So I want to give you a couple lessons on John's approach to science, and I'm sure Bill and Swami and Tyann could add to this, but stay focused. He really hated the concept taking shots on goal. John was really big on, you really want to, science is really hard, what we're doing is very difficult. If a new person came into our research teams or into our clinical development and talked about shots on goal, you could say, oh, you're gone. Because John just really hated that. He was really wanted to stay focused. Hire excellent people and build world-class internal research. There was always a rumor going around that John always had a list on his, about the people who couldn't make the cut in the company. But he was really big on having the right people around him. And one thing John said a lot is, it takes a team of really good people to develop and bring a product to market. But he said it takes one idiot to screw it up. And so he was really big on making sure he always had a world-class people around him. He also said at the same time, no matter how big you are, you're only a small percentage of the research in the world. So he really wanted us to go out there and partner. And John, I don't have a slide of this, but we've all seen this, those who work with John. He would always go to the chalkboard, and he'd draw a big circle. And he said, this is all the R&D in the world. And I don't know what the number was, 200 billion or 300 billion. And then he'd draw another big circle, and he'd say, this is the NIH. That circle would be smaller today, unfortunately. But he'd do that. And then he'd draw a little dot, and he'd say, and that's Pfizer. And that's where Pfizer's doing their work. And think about where we are. If we think we're going to find the best science in our little dot, we're wrong. So what John would want to do, and this is what the team did, not me, but the scientists, he wanted to have the best science and the best internal research. But he really wanted them to go out in the world, go to scientific conferences, go to academia, go around the world, places like Scripps, and meet with people like Pete and meet others, and find out where that best science is, because he wanted to make sure that he had the best science at Gilead, and he was going to bring it in there. So that was part of what he did. Don't do anything internal that can be done better external. That has to do with his partnerships, and I think Tyann probably bear the big brunt of this, where, for example, in manufacturing, John was always big on saying, we don't have to build manufacturing. There's tons of really good, particularly small molecule manufacturing around the world. Let's not make that, that's not going to be our big, game changer. So he wanted to partner that. But he would also partner in a lot of other areas, whether it be in research or discovery efforts and otherwise. Another one that I learned from John, and those of you who are starting up companies, take the money when it's there. And I sit on a lot of boards right now, and we often, and if there's any VCs in the room, I apologize. People are always concerned about dilution and things like that. John's big push was, we always need to have the money to do the research and do the development we want to do. We never, ever want to be in a position where we can't do what we want to do because of lack of resources. So I think one of the things that Gilead did really well under John's leadership was, when we had the ability to raise money, we raised money. And John hated absolutes, never say never, never say must. If you're in a conversation, he always wanted to sort of keep options open, always keep the idea that you're never dictated to do one thing or the other. And so that's just a little about John, just a little science. I'm not a scientist here, so the scientists in the room probably have heard many of these things before, and there's probably another long list of Johnisms or Martinisms that I could probably put here. In terms of John finding the best science, I mentioned that John would go out into the world and seek out the best science. He also was developing personal relationships. So this is a picture of John early on with Dr. de Klerk. That's Eric de Klerk in the middle, and Anton and Holy. So Dr. de Klerk is from the Riga Institute in Leuven, and Dr. Holy is from IOCB in the Czech Republic, or I think when John first met him, it was actually Czechoslovakia. But this is a young John. This is at the Rio Grande. They were at a scientific conference. It was ICAR, I think. ICAR. And this is what was going on here at this time. This is when John recognized that we needed to have the nucleotides. These guys developed the nucleotides. There was a partnership between IOCB and Riga. And John spent years and years building a relationship, understanding their science, and eventually bringing that into, actually it was at BMS originally, convincing these guys to take it back from BMS, who wasn't going to develop it, he didn't think effectively, and then license it to him at Gilead. Small little company based on antisense, but John just had that drive to make that happen. So that's just a little bit of John and his approach to science. And this is the results. We talked about some of this already, but John had a view of simplifying HIV therapy, making it simple, making it tolerable, and making it easier for people to be compliant. That would be the key to people remaining on therapy and avoiding resistance, avoiding breakthrough. First single tablet regimen in 2006 to Biktarvy in 2018. I think if I was to add to this today, I'd put lenacapavir on this. John was really big on the capsid as well. I know that you guys in the room worked on that. Another thing that John would say, and this is represented by this slide here, atripla was a fantastic drug. It really solved many of the problems that were existing in HIV therapy at the time. Its downfall was CNS toxicity, but again, something else John often said is, if we don't continue to innovate and improve upon our medicines, somebody else will. So we always need to stay ahead on that. And then in hepatitis C, we did the same thing, again, partnering, bringing in sofosbuvir originally from Pharmasset, and then developing a whole arsenal of single tablet regimens to treat hepatitis C. Then in terms of the social contract, so the first product that we developed, a part of the single tablet regimen was Viriad, and this is tenofovir disulproxyl fumarate. This was licensed in 2001, and I remember very well John coming to my office and saying, this is literally like a week after the product was approved in the US, and said, we need to figure out a way to get this into places like sub-Saharan Africa, because this is where the disease is hitting the hardest. This is the biggest burden, and we need to figure this out. And what was going on at the time, regulatory approvals were difficult, there were a lot of government problems, forecasting was difficult, healthcare workers, all kinds of obstacles to making that happen. But in John's way, he's just like, it wasn't an if, it wasn't a try to do this or figure out how to do this, it was do this, make this happen. And that gets to my sort of lessons from John on his approach to access. Be clear on the goal, develop a smart approach based on one goal, patient access, so make it happen. Protecting a business and IP is very important, but he didn't want to make that the primary goal. And so I think one of the things that was going on in terms of when people are talking about access is, well, I want to do this, but can we do this in a way that's going to protect our business? And I think John sort of flipped that over, and the other side said, no, we will do this, and we're going to make this, first and foremost available to patients, and we'll take that risk. We'll take the IP risk. We'll take the risk on our business. Get out personally and meet the patients, provider, policymakers, and activists. And I think that's really key. I think that one thing that I learned from John, when you're negotiating with governments around the world or negotiating with activists or others, you find out actually you have a lot more in common than you disagree on. We tend to get into, in the pharmaceutical area and with governments, focusing on price and negotiation of price. When actually what we want to do is the same thing that the governments want to do, the same thing the patient groups want to do, the same thing that the activists want to do. We want to treat disease. So you start there and you start to build those relationships. And you're going to get into price discussions, but find that commonality first. Don't pursue good PR. Do what you think's right. So John was, when I first looked at what other companies were doing in the area of global access, and this still is true today, a lot of them were PR campaigns. And John was very clear, I don't want a PR campaign. One of his sayings, he's actually from the Midwest, was you can't put lipstick on a pig. So if you're doing something that's wrong, just don't try to cover it up with a glossy PR campaign that's, it's like, we're going to save the world and that kind of stuff. No, John wanted it to be real. So he said, don't pursue PR, be honest with what you do, be transparent. If people don't like what you're doing, as long as you feel you're doing the right thing, keep doing it. That was his approach. Don't make decisions by committees. That was a big thing that John said. He said that committees are really good at protection, providing guardrails, but really bad at making decisions, really bad about pointing you where to go. And so in terms of, when I look at our access program, we could have set up a committee that told us all the risks that were out there, all the things that we should be worried about. And John was like, that's great, but we need to, first and foremost, this is what we're going to do, and we take that risk. And use an innovative approach to access. So John, research what you want to do with the problem, come up with a hypothesis, and I'll talk about how that worked for us. Test it, and if it doesn't work or if things are wrong, adjust it and redo it. So he took a very scientific approach in terms of access. So in terms of our access program, I mentioned in 2001, he said, make this happen. We started out with a very simple program in 2003. We said, okay, simple, no profit pricing. So we won't charge a profit in the poorest countries of the world. We'll do a direct shipment to the countries. and we'll bypass all these regulatory burdens. We thought this would be easy. That was our hypothesis. It didn't work. By 2006, we got about 30,000 patients on treatment out of the probably 30 to 40 million who needed treatment. Complete failure. But John recognized that, and he said, okay, let's figure out what we need to do differently. So the first thing that John did was he separated out what we're doing in access from our commercial business. He really felt that we needed to have a focus that's not looking at the protection of profits, protection of the commercial business, just an operational component of the company that's focused on getting product to patients in poor countries, establishing distributor networks, medical affairs functions, and all those things. The key thing that John did in 2006, and I think this is part of the innovative approach, is we talk about partnering. And we said in 2006, is anybody doing a good job of delivering medicine in, say, sub-Saharan Africa? And the answer is yes. There were people doing a good job of this. And it was the Indian generic companies that were. And so John said, well, what are they doing that we're not doing? But more so, why don't we just work with them? If they can do this, why don't we we can't really model our business after them, but why don't we work with them? So in 2006, we launched a generic licensing program. And what we did is we took the world and we divided, looked at the poorest countries. So you see sub-Saharan Africa, you see India, parts of Eastern Europe, and parts of Latin America. And we wanted to recreate a generic marketplace. And so the hypothesis was we can't just have one partner. We can't have two partners because that's not the business model that has created the value through the generic model. We need to basically open this up so we could have a true generic marketplace. We'll have contracts with them to protect our IP and limit where they can sell, so they can sell into these countries. We'll have a low royalty, 5% royalty. We'll allow both drug product and drug substance. We can't just limit them to drug product. So really allow them to do this the way they do it and have no controls over how they manufacture, their science, allow them to do this way. And the idea was that it would create a generic competitive marketplace for our products in terms of to really allow the generic companies, the Indian companies, to do what they do best, which is large-scale manufacturing, their chemistry, and their distribution. And what we saw was from 2006 to 2018, the price of the product dropped from $17 in 2006 to $1.80 in 2018. For a triple, that's 26 and 370, or for Truvada, 370 in 2018. And it's actually gone down further even since then. So the experiment worked. We actually were able to bring it down. And that made a huge difference for patients. So if you look at the same thing that went over that period of time, we went from that roughly 30,000 patients early on to 12.5 million receiving drug in 2018. And that number has grown since then and continues to grow. In this place. And this is just showing sort of the process of this, the license agreements in 2006, the various milestones, the way 30,000 patients in 2008. And then we actually have other programs for donations and other things along the way. In terms of what we've seen today, dramatic decrease in the number of people who die from HIV, dramatic decreases in the spread of the disease, largely because treatment is prevention. Women and children still remain a big problem in the developing world, but that is something that John was working on largely through prevention programs. So just a huge amount of progress. I'm obviously not going to read this, but I think John would say there's still more to do, and I think we're seeing that through his science even today as it continues. Partnering, I mentioned partnering. I just want to show you a couple slides of John. This is a program we had with the Chinese CDC. They had a problem with HIV, largely due to blood transfusion, blood donation programs in China. The CDC didn't have the money or the funding to treat the patients, and John said, hey, we'll work with you. So this is us in China, that's with the CDC, signed an agreement to basically donate tenofovir to their program to treat the patients. And what's remarkable is the CEO, like John, actually spending the time to go to China to meet with the physicians, to meet with the government and make that happen. That's just a little bit about who John was. This is John in India. John loved going to India and meeting with our Indian partners. It was just fascinating. I said how John was very personable. My best memory, or one of my best memories of John is we're in, I think it was in Delhi, I can't remember, we're meeting with then Matrix, I think they became another company, but one of the Indian companies, and we're sitting on a street corner, drinking beer, eating peanuts, and John just talking combinatorial chemistry with the Indians. It was just, that was like totally John, but he loved doing that. He loved meeting with patient advocates. This is in Georgia. John became very close with some of the physicians and leadership in the scientific community of the country of Georgia. That translated into an elimination program where we donated hepatitis C products to Georgia, really to show that you can actually mobilize a country and eliminate hepatitis C with these products, and trying to encourage other countries to do that. We did the same thing in Egypt as well. And the person to the, I guess that'd be John's right, but the next, between John and me, is a patient advocate. He was so pleased that he was finally cured of hepatitis C. He was actually an artist as well, and there's actually a piece of artwork in Gilead that he donated to us. That's pretty fantastic. But that's just a little bit who John was. Again, this is in Georgia. John would travel to the place, meet the patients, meet the governments. I felt that really was what allowed John to see the solutions that needed to be done. He would not just sit in his ivory tower in Foster City and not really understand what's going on in the world. And this is actually the scientific leader in Georgia, a guy named Dr. Tenges. This is in at a dinner, and they're probably talking science there, or they might be talking about the latest iPhone, because that would be something John would do as well. What would John say today? Basically, don't develop Me Too drugs. I think we've talked about that. Others have said that. John had a simple saying we say. He really had a lot of discomfort with the industry. He said they just developed Me Too drugs. He said they just market as blue soap, red soap, purple soap. John was not into that. He wanted to have drugs that made a difference. Be innovative and challenge the patients, that address the challenges that they face, including the systems that they work within. As I mentioned before, be a strong advocate for intellectual property, take risk, measure success in broad patient access, not in commercial return. John was really big on talking about how many patients received his medicine, how many people we've cured, how many disease we've prevented. He really wasn't that proud of talking about profits or revenues. And take time to understand the true patient population. As I said, he would get around and really meet with people and get to understand it. So I hope you've learned a little about John, got to know him a little better, understand his commitment to global access. It was a little bit more condensed than last year, but I would say that I just really miss John, and just so lucky to have been able to work with him and learn so much. And he was a very good friend, and I'm glad that you're able to do this for him. Thank you.
Great, thanks a lot. No, that's good. Thanks a lot, Greg. That was a really terrific overview of John and his thinking and his impact. As we said, John was not only an amazing scientist, but business person and humanitarian. And I think, I'd like to thank Nick and Stuart for their talks, because I think they really underscore one of the themes that Greg brought out, which is work on really important problems that have a huge impact and transform science. And I think both of their talks really kind of underscored that theme, which was so much a part of John. You can also, as I think Stuart alluded to, you can tell a lot about a person, not only by the person. I was really fortunate enough to work with John in the nonprofit world, not Gilead, but it was a wonderful experience. But you can tell a lot about a person also by the people they surround themselves with and the people who they work with. And I think, as much as I appreciate John, I also have, through John, gotten to know all of his colleagues and coworkers at Gilead, and they, like him, I think, share many of the attributes that we've talked about today. So we're really fortunate also at Scripps to know John's extended scientific family. And finally, I'd like to thank you for participating in this memorial to John. We continue to think of him all the time and about what John would say. Why don't we can adjourn to a reception outside. Thanks again.